I am Mohammed Abdul Karim, a professor of fisheries and aquaculture, together with Dr. Zikrullah A. Yusuf, specialized in ecology and fishery, followed by Mr. Dongwera K. Sheltu, my mentee, specialized in animal health and production in the area of reproductive physiology, and lastly Dr. Aisha Bala, specialized in agricultural extension, all in Abubakar Tafawa Balewa University, Bauchi, Nigeria. We are going to present a fishery proposal titled Developing and Advocating for Techniques of Production for Sustainable Local Chain Supply of Spawning Hormones from Fish and Mammals. Background of this research: adequate supply of quality fish seed, that is fingerlings, has been a major problem in the attempts by many fish farmers to culture fish, and is a fundamental prerequisite for large-scale fish production. Today, many fish farmers in Nigeria are unable to produce their own fingerlings due to, number one, financial, two, technical, number three, logistics demands, and have to rely on the government. Induced spawning guarantees a reliable source of fish seed, that is fingerling, in quality and availability. Today, prominent synthetic hormones brands like Overprin, Overtide, Ovulin are in use, but still not readily within the fish farmers' reach. In this research, natural and synthetic hormones are to be evaluated for their efficacy in respect to fertilization, hatchability, and survival rate, while taking into cognizance the economic use of both resources. If you rely solely on the foreign goods, that would be a problem. Take, for example, during COVID, everything stopped, marketing stopped, supply of these synthetic hormones also stopped. I've been advising and advocating for utilization of our available resources in the country, so that this problem will never arise. The African catfish, that is all fish with bubbles, possess whiskers that are called catfish. Clarias garapinos, Clarias angularis is a dominant in freshwater environment, including lakes, rivers, and dams, and is mostly preferred in aquaculture, that is in fish farming, due to its ease of culturing. It has a fast growth rate, high resistance to disease, tolerance of wide range of temperature, low dissolved oxygen, as well as high salinity level, and most importantly, high commercial value, and it is a nutrient dense food. In Africa, the demand for catfish has increased as food, and it is used for control of overpopulation of tilapia in ponds, as a bait for the Nile perch elephant fish. Most farmers depend on fingerlings collected from the wild. However, in the modern agriculture competitive world, this practice cannot be sustained due to the fact that most fishes are seasonal breeders, and that there is always a scarcity of stocking materials called fish seed when it is not breeding season. African catfish are the most common type of fish species for aquaculture production in Nigeria. The major concern of any seed production in the hatchery system is to produce the maximum number of quality fingerlings from the available brood stock. And one of the requirements for fish breeding is establishment of hatchery unit, where seed production and multiplication rely on the use of hormone, be it natural or synthetic, to induce spawning of artificial fertilization, incubation of fertilized eggs, and subsequently feeding live fish, followed by dry feed. This research seeks artificial to exploit the use of synthetic, example overprint, and non-synthetic human mature cows hormones, and also as a tool for breeding catfish. African catfish fingerlings have high demands, and thus their price keep on skyrocketing. Hatcheries focus on management based on existing protocol on fish breeding using synthetic hormones. The cost of this hormone is high and not readily available, particularly in the rural area. This research is exploring the use of non-synthetic hormones from urine of pregnant mammals. Mammals mean any animals, anything that can breastfeed. Human, mare, cow, sheep, and goat for breeding catfish as substitute to synthetic hormones. Also, there are non-established local network for synergistic relationship among the health center, animal marketplaces, animals, and fish farm to ensure supply of input from mass production of induced fish spawning hormones and supply chain of output. The use of pituitary glands of gravid Clarias garaminus as it is normally practiced in much of our fish farm for induced spawning has been established, but the fish are usually sacrificed. They are killed without or with little market value The table fish are the potential sources of this pituitary gland. However, techniques for extraction of fish glands without affecting the market value of the fish are the gap this research will be exploring. The research will explore the gap through advocacy. Our research questions are: A. Can non-synthetic hormone in urine of pregnant women, mare, cow, sheep, and goat be purified, extracted, and stored for commercial usage? B. Can less invasive technique for extracting pituitary gland from tableside catfish without affecting its market values? C. What is the percentage hatch, mortality, and survival rates of fry from catfish induced with the non-synthetic hormones obtained from urines of pregnant women, mare, cow, sheep, and goats compared to that of synthetic hormones? D. Can there be established model local network for synergistic relationship through advocacy for sustainable supply of urine of pregnant women, mare, that is horse, cow, sheep, and goat for health centers, farm, animal marketplaces, and veterinary clinics in Bauchi and Nigeria at large? Our research objectives are: A. To utilize the urine of pregnant women, mare, cow, sheep, and goat in induced breeding of catfish, Calarias garipinus. B. To extract hormones from the urines of pregnant women, mare, cow, sheep, and goat in induced breeding of catfish. C. To develop less invasive techniques and simple tools for extraction of pituitary glands from tableside catfish without reducing its market value. D. To develop less invasive techniques and simple tools for extraction of pituitary glands from table-sized catfish without reducing its market value. I've already mentioned that. E. To develop model of local networks for synergistic relationship and protocol for sustainable supply chain of urines of pregnant women, mare, cow, sheep, and goat for mass production of induced sperming hormones through advocacy among farms, animal marketplaces, and veterinary clinics in Bauchi and Nigeria at large. Literature review. Under this literature review, we are going to discuss two aspects: utilization of non-synthetic hormones for induced breeding of fish species and the place of Calarias garipinus in aquaculture production in Nigeria. According to the earlier work of Lam et al. 1983, pregnant woman urine contained hormones gonadotropin. Generally, this hormone gonadotropin promote egg maturation of reproduction of fish species, which also controlled by several factors such as sex steroids in the regulation of reproductive processes. These reproductive impregnants are controlled through the brain pituitary gonadal axis. The brain is stimulated by environmental cues such as water rise, temperature of the water, feeding, rainfall, and photoperiod, in order to release gonadotropin releasing hormones. Ovulation and spermiation are affected as a result of sex steroids that are being produced. Induced spawning by using HCG, human chorionic gonadotropin, produced from urine of pregnant women, were used in a research conducted by Mammals and Sujin. Urine from 2 to 3 months of pregnancies were recommended by these authors for HCG extraction, that is human chorionic gonadotropin extraction. The dosage of 200 to 800 IU per kg of sperm are appropriate for clearance, according to the authors. But the concentration that were produced of HCG by the authors was 5,000 IU per vial. That is, they were able to produce 1 liter, they were able to collect 1 liter of urine. 1 liter of urine collected were used to produce 5,000 IU of high HCG. In our research, the urine of cow, pregnant cow, mare, and human containing crude hormones not extracted were injected. That means crude hormones were injected into 3 female catfish, each at a dosage of 2 ml per kg of light body weight, light body mass. While synthetic hormones, ovaprine, was injected into a female catfish as our control at 0.5 ml per kg. Shaltu, Dungwera, Abdul Karim Mohammed, Manchawapi, Zukrulai, AY, reported that both synthetic hormones, ovaprine, and non-synthetic hormones in urines of pregnant cow, mare, human successfully induced spawning of Clarias garapinos. The authors concluded that pregnant mammals' urine can induce breeding in Clarias garapinos as an alternative to ovaprine. The major bottleneck in culture of Clarias garapinos is seed production, as the species is relatively difficult to reproduce in captivity. We can now take the place of Clarias garapinos in aquaculture production in Nigeria. Nigeria is blessed with ruminant and monogastric animals. Scientists should be encouraged to utilize pregnant animal urine for induced breeding of catfish, as observed by Adebayo 2008. In Africa, the fish farming community mainly use natural pituitary suspension for catfish-induced spawning. This method has proven to be highly reliable compared to synthetic hormones analogs. Ovaprene, ovulin, and the rest. In terms of cost and availability, as reported by Richter et al. 1995, catfish does not spawn in captivity even under favorable farming conditions. As a solution, several hormone-induced breeding techniques were developed for Clarias garapinus farming, including the one we are advocating: utilization of pregnant human and animals' urine for induced breeding of catfish. Theoretical framework. Most farmers depend on fingerlings collected from the wild. However, in this modern and agriculturally competitive world, the practice cannot be sustained. African catfish are the most common type of fish species for aquaculture production in Nigeria. The major concern of any seed production is hatchery system required for fish breeding, which relies on the use of hormones, be it natural or synthetic hormone. Synthetic hormones are very expensive. Their availability is not reliable. Unlike natural, which is readily available. The recent advances in the science of fisheries culture technique have resulted in an increased understanding of the crucial role of endocrinology, that is study of hormones, especially ductless hormones. In aquaculture, pituitary gland secretes the gonadotropins such as follicle stimulating hormone and luteinizing hormone. Luteinizing hormone causes luteinization in females and promote the production of testosterone in males. Luteinizing hormones, as earlier mentioned, promote the production of testosterone in males. The gonadotropin hormone, which is one of the hormones produced by the adenohypophysis of the pituitary, stimulate the production of eggs and sperm. Gonadotropins regulate the formation and maturation of gametes indirectly through steroid hormones produced by the gonad itself, with many numbers of variation in patterns in the pattern of the events, depending on the seasonality, cyclic maturation, and even location of the freshwater body. This research seeks to explore the comparative use of synthetic and non-synthetic hormones from women, mare, cows, and got or pregnant in the first trimester and fish, and also to localize the supply chain of non-synthetic hormone in Nigeria. It means we are going to produce this hormone, package them in vials, for an onward supply to interesting fishermen and fish breeders in the country. This research will be conducted in Abubakar Tafawa Balewa University Research Fishery Farm. The hatchery unit of the farm will be upgraded for this research. Catfish, Clarias garipinus broodstock, will be obtained from a standard hatchery farm in Jos and Bauchi states. Gravid males about 6 and females 6 catfish will be used for induced breeding. The broodstock will be fed 5% of their total body weight and allowed to acclimatize for at least a week. Non-synthetic hormones will be collected in a sterilized bottles under accepted measures from pregnant women of about 3 months pregnant at Abubakar Tafawa Balewa University Teaching Hospital, Bauchi State. Pregnant cow urine will be obtained at Fulani settlement within Bauchi local government area very early in the morning. 3 months pregnant horse mare urine will be obtained at polo field stables through the assistance of the attendants of the unit. Synthetic hormones will be purchased from fish store within Bauchi and Jos, Plateau State. Precipitation methods as described by earlier work of Tangru Pinos will be followed. Urine from 2 to 3 months pregnant women and mare and cow will be used. Human chronic gonadotropins will be extracted by precipitation method. Precipitation with acetone at the rate of 1 part of urine and 2 parts of acetone will be shaken for about 10 minutes and will be kept at 4 degrees centigrade. The clear solution will be poured out. The precipitation part will be centrifuged at 4,000 revolutions per minute for 10 minutes and filter through Whiteman paper. The precipitate will be kept at 4 degrees centigrade until used. The concentration of human chronic gonadotropin can be measured in a standard laboratory by double quantitative human chronic gonadotropic radioimmunoassay. The crude human chronic gonadotropin will be freeze dry for 20 hours. The crude human chronic gonadotropin will be kept in vial with 0.01 gram per vial. Statistical analysis. Mortality and survival rate will be determined according to the method described by Agboegbu, 1985. Weight gain and final weight gain and specific growth rate will be determined following the standard methods. Effectiveness of non-synthetic hormones from fish, pregnant women, mare, cow, sheep and goat will be established. Techniques and protocol for mass production and model for sustainable localizing non-synthetic hormones supply chain in Nigeria will also be established. Innovation. Methods and techniques of mass production of non-synthetic hormones for induced breeding of catfish Clarias garapinos and its sustainable local supply chain in Nigeria will be established. Tools for less invasive extraction of pituitary gland from stable sized fish will be fabricated. Sensitization program among pregnant women to consider donating cells urine as similar to blood donation and cells will be carried out. And synergy between aquaculture industry and maternity clinics will be established. This will be achieved through patent registration of the method and training and retraining of interested persons on the established protocol from this research. Thank you so much.